Hey Gracie, did you make sure to pack a lunch?
Worrying about yourself.
What I told me to choosing my clothes and going to the bathroom.
Has subscribed to you. Who the? Somebody named Jurassic Parkour just decided to subscribe to me. I'm going to safely assume that that's a student. Safe assumption. Nothing. Somebody with the YouTube name Jurassic Parkour.
Yeah.
All right. It's live, like I'm here. So y'all just start getting on. Make sure y'all can hear me. Remember, if you're on your school Chromebook, you won't be able to comment and ask me questions.
My iPad thought you said hey Siri.
So if you want to be able to comment, to ask questions, get on a personal computer, iPad, something like that.
Remember that computer piece.
Oh, yeah. It's no like.
Yeah. Try not to move around too much.
I have to move around. I can't just sit here. This just sitting doesn't work for me. Start getting on, people. Here's the thing. Some people don't watch the review. They like the live because they're like, I don't really have questions. I'll just watch the video afterwards. Okay. Whatever floats your boat.
Should I continue drawing buildings or should I draw like the
Background.
background?
Like a park. Trees. Like you're looking, like it's an aerial view and there's trees and like a park behind it. Like Central Park pictures and stuff.
Oh, this isn't going to be realistic. This is my art. I don't do realistic. I literally made it.
Okay, then make it a unicorn playground.
No, that's okay. But I get what you're saying. That's something like that.
Per usual, guys, Gracie's doodling. I mean, come on. Do we expect anything else from her? No. No, we don't. Because she just doodles. She doesn't pay attention.
I do pay attention.
Whatever.
Bye, Presley.
Alrighty.
Ooh, this could look like trees. Maybe I could make it like slightly realistic.
You would think having your biology teacher as a mom, she would take advantage of
I have. I don't need any more.
that she did. Ooh, y'all didn't just hear, but she just said, I don't need any more help. What?
That's not what I
Oh, what did
said.
you say? I don't need any. You said I don't need any more.
Advantage.
Oh, I think that's even worse. She said, I don't need any more advantage.
This isn't looking like trees.
Gracie. Gracie.
Oh my God.
Riley says, so I know the structure of proteins and nucleic acids, but I don't know what the elements mean and what type of questions you'll ask. Oh, no, you didn't, Grace. Nico just said, Gracie is advanced, Shrimon said. Yeah, Shrimon,
This
I'm
isn't
not so
looking
sure
like
about that.
trees.
All right, so we've got a few people on here, so I'll go ahead and get started. Alrighty. Okay, so Riley, the elements that we're talking about is just what they're made up of. I don't stress it so much. The elements are called schnapps. It's a way to write it. I'll just write it down here. So keep in mind, guys, this whole unit is about biochem. It's about the molecules that make you, and we need to put them into their big categories and discuss each of them just a little bit. Obviously, there's more to it than we can discuss in this kind of class. So we've talked about the 4 major ones: carbs, lipids, proteins, nucleic acids, right? You cannot dismiss the first 2. Even though they were on the last test, there will still be like references to these. So you can't forget about them. Especially you can't forget them because, well, it's final exam, star, whatever, okay? So carbs and lipids, proteins, nucleic acids. Obviously, we're going to ask more questions about these 2 than we will these 2 at this point. But here's the thing. Schnapps, it's a way to remember the major, not all, but just the major elements that make up these biomolecules. So it's schnapps for carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Just a tricky, unique way. Obviously, because these are all called organic compounds, they all have to have carbon. They all have to have hydrogen because carbons are going to bind. And then they all have oxygen. Then the N and P and S is what just makes them unique. So I'll just kind of write them off to the side here. Carbohydrates just have carbs, carbon, hydrogen, oxygen. Your lipids just have carbon, hydrogen, oxygen as well. Sometimes they'll have a phosphate added onto it for phospholipid.
You drew a spaceship.
Oh, okay. Good job, Gracie.
Technically.
Proteins have carbon, hydrogen, oxygen because all of them do. But then they can also have the nitrogen because that's how you identify a protein is the nitrogen. They can have a phosphate and they can have a sulfur because that R group on your proteins. And the nucleic acids have carbon, hydrogen, oxygen. And then they have a nitrogen and they have a phosphate. So it kind of just builds on top of itself. CHO, CHO sometimes P, CHONPS, and then CHONP. It's just a way, schnapps is a way to remember, Riley, the elements that make them up. Okay. But again, I don't stress the elements too much. Miss Martin may stress it. So y'all just need to remember the proteins have everything. The nucleic acids do not have the sulfur. That's the one thing that distinguishes between these 2. Okay. All right. So as I stress with my students, you need to be able to do this chart. If you can fill out this chart pretty well, then you're doing good on the organic compound part. Okay. Do you know the monomers, the structure, the function, and the examples? Like if you can fill this chart out from memory, then perfect. Then you don't need to really stress this too much more, okay? So the main ones we need to focus are proteins and nucleic acids, right? Amino acids build these. Amino acids get linked together with a kind of bond called, Gracie?
Amino acids get called peptide bonds.
Peptide bonds, she did! Shrimon, Gracie L advanced, yay! Okay, so amino acids bond together, peptide bonds. And remember, you have to recognize this nitrogen. That's your amino acid. This is one amino acid. Many of these bind together to form your polypeptide, your large protein. All of them are going to be unique because of that letter R. Remember, that's just a placeholder, just like in a math class. Examples are anything. Guys, proteins are everything to you. Proteins are everything. They are your transport. They are your cell membrane. They are your support and structure. They are your immune system. They are enzymes. What else did I put on there? I think that's all I put on there. And then, of course, they're your hair, your skin, your muscles, right? All of that stuff. Meat. Okay. We're not even going to go there. Gracie asked a question about, wait, when I think protein, I think meat.
Well, James was talking about
okay, guys, meat is the muscle of the animal. When you're gnawing down on them, you're eating their muscle. That is their protein. Okay, and then just understand enzymes, right? Be able to recognize enzymes. Enzymes always end in ASE or SIN. So catalase, amylase, pepsin, trypsin. All right. And then on the nucleic acids, be able to identify that they're made of nucleotide. I can tell you right now, you will try and tell me that an amino acid builds a nucleic acid because you go with that acid. Don't. Go with the first word, the first part of the word, nucleic. So this thing is a nucleotide. That is what we call a nucleotide. Many of these monomers will build together to make your DNA, the stuff that makes you, your inherited material, your genes. Okay? You need to be able to draw a nucleotide and label it. Okay? Circles are always, what are circles, Grace? Phosphates. Pentagons or hexagons are always?
Oh, carbs.
Carbs. Good job. Man, she is big brain in this right now. And then this is a box.
It's a base.
It's for a base. You don't need to worry about what that is yet.
No, you don't.
We'll talk about it later. But you need to be able to draw this. You need to be able to recognize this and understand which ones they go for. Protein versus nucleic acid. Okay, let me look. Levi says, is it called something different when two substrates bind? Or is it just a product? Oh, yeah, no. The substrates, so when substrates come together and they're just forming a product. Now, remember, the processes that build things is called dehydration synthesis. Like, we call that process that name. But it doesn't matter if you're grabbing 5 substrates, 15, 2. When you put them together, they form a product that is that chemical reaction. Now, what you're probably thinking is, if you put things together, what kind of reaction? That's an anabolic. Versus if you break it, if you break one substrate into products, into separate products, then that's catabolic. So those have different names, which we'll look at on the enzymes and stuff. Okay. Yes, reactions. Good job, Shriman. Okay. Guys, if you can do this, you're good on that part. All right. Questions on the chart. Okay. How to draw a nucleotide, how to identify an amino acid. Okay. I know in Miss Martin's class, she stresses what this is called. That's how you identify it, because the nitrogen with 2 hydrogens is called an amine group. It's an amino acid, that's why. So an amine group, and on the other end would be an acid. So the whole thing is called an amino acid. Okay. All right. No questions on there. Let's go ahead and flip over to the reactions. Look at that. I have them right. Yeah, I have them right upside. Okay. All right. You need to be able to draw this on your own. If you have a scratch piece of paper right now or something, I suggest you be getting it out and practicing along with me. That's the only way, guys. I cannot stress this. This is the only way you get better at studying, is doing it just repetitively. Do it, draw it, write it. Do it in my class. Why do you think you get better at math? I swear, people think math's the only one that you actually need to do practice. You need to practice this stuff in any class. Okay, I just typed out like 4-sentence question about the amino acid identification, and then you answered it. Good. All right, so your graphs. These graphs simply describe a chemical reaction from beginning to end. So this is time happening. The actual reaction is taking place from this direction there. And then the Y is just the amount of energy being used. Okay.
Pick a color.
She ain't paying attention for
I am
anything.
paying attention.
Purple.
I did purple, actually.
Okay. So time versus energy, or time, not versus, but time and energy. All right. You need to recognize the difference between these two graphs. One goes up, one goes down. That's like the biggest difference. But what this is telling you is this one, we're going up, which means we are actually building something. Okay. So you can kind of, things to help you remember that this is building. We're going up, like we're building a building, right? We're making it taller.
I'm drawing buildings.
Oh my God, Gracie, why? She's drawing buildings. Okay, so if you can recognize this graph and the fact that it goes up as you move forward, like you're building something, this is us building polymers, us building dehydration synthesis, us making our large starches and stuff like this. That's this chemical process. You growing is this, okay? So we have to start with reactants. We have to end with products. So that's the first part you need to be able to label. You need to be able to identify a reactant versus a product on this graph. What's the reactant and product? Anything. It can be anything. Your reactants can be 2 glucose molecules, and your product is a type of sugar called a maltose. Okay? It can be anything. This is A plus B equals C. Right? Whatever the guys, I could be talking about a cake. This is your eggs, your sugar, your flour. This is your cake. You built it. Right? So we've got to go up to build, like a building. We're flowing up. Now you've labeled the reactants and products. Now we also need to label that hill. And that hill needs to be identified as the activation energy. Okay? So from the reactant to the top of the hill is the energy. And yes, I write it E sub A because energy is the letter E, but we need to identify this as activation energy because it is unique. Not all energy is the same. So an activation energy is how much energy does it take for this reaction to get started? Like how much energy is it going to take for it to get going? So for this reactant to become this product, whoa, that's a lot of energy. Well, yeah, because you're building something. Guys, you're sitting there with your Legos, block after block, monomer after monomer, clipping them together, dehydration synthesis, to build a huge Death Star, okay? Mm-hmm. Whatever it is. So you're clipping all that stuff. That's a lot of energy, right? That was a lot of time and effort to build that. That's what you did here. You put a lot of energy in, and voila, you made something. Okay, you built it. Now, just because you can label these parts, now we also need to label what kind of graph these are. The terms associated is energy, so endo or exothermic, and then also what's happening to the bonds. Are we building bonds or breaking bonds? Right? Exo always goes with catabolic. Endo always goes with anabolic. That's how it works. In the whole process, it comes down to understanding the chemistry behind it. But if we need to build bonds, then we're going to have to gather a lot of energy. Energy. Because you're about to do something like crazy big, right? Death Star. So we've got to build and put energy in. So here, if we're going up, we're building, then this becomes anabolic. Okay. And then anabolic is always associated with endothermic. All right, so anabolic and endothermic, those go together. If you can remember that those go together, then you're good. Now, how do you see it on the graph? If the reactants, if you're thinking of energy, just like a y-axis, okay, just like you're building a graph, this reactant is probably at like a level 100, while these products are at a level 400. We had to make a product with 400 units of energy, but we only had 100 to give. So we had to put energy into our system to make it work. That was the oven helping to bake your cake. That was you, blood, sweat, and tears, building that Death Star. All right? So endo and ana go together. You've got to build, you've got to put energy into. Guys, prefix, suffix, save lives. Endo means into. All right, we've got to put into, and we're building the bonds. Now, if you know one, then you should be able to recognize the other, right? I think, personally, more than anything, I think this one is actually an easier one to remember because we start high, we end low. Why was that funny? Oh, okay, Gracie. Detour. All right, so we start high, end low. Here's a couple of things. the words associated with, I feel like just really go with this one. But we're going to label the same way, right? We always have to have our reactant on the far left, and we always have our product at the end. So reactants, products. But now look at our energy, activation energy. It's different. It always is where the reactant is to the top of the hill. So our activation energy is smaller, but there still has to be one. We still have to have something to get the process started. So our E sub A is smaller for this reaction, but it's still there. Now, words associated with it. We start up at a high energy and we go down because your prefix word for down is what? Cata. So this is catabolic because you're going down in this graph, down cata, because we're going to break bonds down. We're going to start, so all your blood, sweat, and tears into that Death Star. Doggo, bad doggo comes in and destroys it, okay? All those bonds get broken and your energy gets released, right? So this becomes exothermic. We had 400 units of energy. We only needed 100 in the product. So 300 units of energy got lost. Chemically, what you see, guys, in an anabolic endothermic, your system gets cold. In a catabolic exothermic, your system gets hot. This is light, this is fire, this is maybe bubbling. Over here, this is freezing. Okay? So you have to be able to recognize and fill this out and label anabolic and catabolic, exo and endo. But they go together. This goes with this goes with this. All right?
Yeah.
Next piece is, on this graph, be able to show me how an enzyme affects it. That's the last piece of this whole puzzle, are enzymes. An enzyme, guys, an enzyme's job is to do what? See if you can answer it.
To like lower the stuff on the graph, the amount of energy that needs to be taken, the amount of energy that needs to be used for the reaction.
It does what with it?
It speeds it up.
Ooh, see, that is something that y'all keep trying to say. It speeds up the rate by lowering the energy needed.
Speeds up the rate. Okay.
Okay. It speeds up how fast your reaction's going to take. Speeds it up, makes it quicker. So you don't have to spend as much energy. And because of that, you're going to lower that activation energy. So on the graph, you cannot change. Let me see if I have a different color pen. Hold on. I do. Okay. Guys, you cannot change where you start and end. Okay? You still start and end where your reactants and products are. An enzyme cannot change that energy. All an enzyme can do is change the energy of the hill, making it lower. Okay? Lowering that energy. And because that hill isn't as high, you get the reaction done faster. Okay? It is, oh, Shrimon beat you to it. He actually said it before you did, to lower the activation energy. Okay. Because, guys, and I use this example all the time, and I hope this example kind of makes sense to you. It is the difference of you walking to school. That's going to require a lot of energy on your part, right? So your home to school, reactant to product, right? Just a way to associate it, okay? Versus home to school via a car. You're definitely going to get there faster. And did you personally have to use as much energy? No. That makes so much sense. Oh my God. That's what an enzyme's doing. Your car was the enzyme. It got you there faster, but it didn't change where you started and where you ended. Just got you there quicker. Okay. All right. So understand how the enzyme affects and be able to draw it just like this. It lowered it, but didn't change where it started and stopped. Okay. You ready for the last piece of the puzzle? I don't think this one's as bad, guys. I really don't. Really, really don't. All right, last piece of the puzzle. If I asked you to draw an enzyme, Gracie, draw an enzyme. All right. This is her enzyme. Does your enzyme look like her enzyme?
No.
Probably not. Is that okay? Yup. When I draw enzymes, I normally draw this. Woohoo! That is hilarious that actually worked out just like that. I did not mean that to work out. That's perfect. Okay. Okay. So it doesn't matter how you draw your enzyme. An enzyme, it looks like a mushroom. It does. An enzyme is just a big fat protein, right? A big glob protein. So I'm going to use my enzyme on this picture only because hers actually fits perfectly into mine as the substrate. Okay. So here's things to keep in mind. Okay. Every enzyme is a lock and key. It is a perfect puzzle piece. One enzyme for one substrate, guys. That's it. You cannot have more than one substrate on the enzyme, yes. But what I mean is only those fit it. It's a perfect puzzle piece. You can't all of a sudden go grab one and try and make it fit with that enzyme. If it's not meant to be it, it can't. Okay? Or an upside down elf match. Upside down. Okay. So it is perfect puzzle piece, an enzyme for one substrate or one set. Oh, shoe match. There you go. Elf. Oh, elf shoe. Oh. Okay. All right. So in this example, you've got an enzyme. I don't care how you draw it. It's an enzyme. Recognize an enzyme has to have some kind of indentation to it. Yeah. Shrevon, I didn't know. I was, it's late. Okay. All right. You have a spot. That spot is, and you have to be able to label all these parts, guys. The spot that is open is the active site. Now, I can tell you that is going to be something that you'll flip. The active site here, and here's what you're going to accidentally say. You're going to call it an activation site.
That's on the graph.
And the activation's on the graph. Or you're going to call on the graph active energy. I don't really have a way to help you make sure you keep it straight. Except for if this helps. When you think of enzymes, think of they are the thing that are keeping you alive because they themselves are very active. So they have an active site. Okay? Active enzymes have an active site. Does that help? Active enzymes have an active site.
And activation.
Okay? And their job is to lower the activation energy. All right? Okay, so active site, substrate binds. So what you'll get is this perfect little enzyme, and now it's perfect little substrate binding directly at that active site. At this point, guys, an enzyme is going to help on either reaction. It's going to help build or it's going to help break. And Levi, I think that's what your question was. If this is one substrate, if it's one thing, then it's going to more than likely, it's going to show that it's breaking it, right? So that's going to be a catabolic reaction it helped with. Or this could be a situation like this, where this enzyme has two active sites because it grabs two substrates and its job is to take those two substrates, bind them together, and release it as a product. And so this would be an anabolic reaction because it's building something. So like this would be an amino acid. That would be an amino acid. At the end, protein. Versus this was sugar, or not even sugar, this was starch. And we're ultimately going to break it into its pieces of sugar. Okay, this was the cracker into sugar versus your body's going to take the sugar you just made and build hair, build cell structures or something. So this would be. So this is building, taking something and making it. So this is anabolic.
Right, yeah.
But ultimately, guys, all I need you to be able to do is look at a picture and say, hey, there's the enzyme. Hey, this is an active site. Hey, these are substrates. Okay. I need you to just label those parts. Did you say something about a question or two about carbs and lipids advice? Yeah. I was just letting y'all know I did end up putting a question on there referencing carbs and lipids. Just referencing them. You should still be able to you should know if you can fill out the chart, you're fine. Okay. It's not an in-depth thing. I couldn't get it to work on the app until Shrimon shared it with me. Also, I'm Toby. Oh, hey, sweetie. I couldn't get it to work on the app. What app? I'm Burke, or am I? I don't know, Burke. Okay. So anyways, take the enzyme, take the active site, right? You need to be able to label that. So just one question. Riley? Yes. YouTube app. Oh, like the YouTube app on your phone? So are you on the YouTube? Like, is that how you're able to comment on using your cell phone? Is you're on the YouTube app and that's how it works, Toby? Okay, good deal. So you can't comment unless you're on the app. Awesome. Thank you. I will pass that on to people for the future. So just making sure. Okay. Can substrates be proteins? Oh, yeah. Substrates can be anything. A substrate is a reactant, a chemical reactant somewhere in your body. And to help it move to whatever it's supposed to do, yes. Everything in your, pretty much every single reaction in your body has to be helped by an enzyme, or else we're going to be using more energy than we should be. And we don't have that. Like, we just don't have that kind of energy, guys. Okay. Yeah. And so chemical reactions everywhere, enzymes everywhere, every molecule in your body is going to be helped at some point in time by an enzyme. That's how important they are. Okay. All right. So be able to draw that, right? Now, competitive versus non-competitive inhibitor. All right. Guys, all that is there are chemicals in our world that can interfere with the job of the enzyme. That's what your lab did, right? Some of you had an acid, some of you had a base, some of you had a salt, some of you had green tea because that actually did it too. And so all of those things can interfere with how the enzyme works.
I'm giving the chair.
Yeah, in just a second. And then here's the other thing. Like, it's not just, things can be competitive and not competitive. Guys, in your body, things can go all kinds of cray-cray, all right? Your pH can be thrown off. And if it is, enzymes aren't going to work. Your temperature can be thrown off, enzymes. Salinity can be thrown off. If those things, if your homeostatic condition is not right, your enzymes will unfold. I showed y'all in class. Let me grab. Oh, I don't want to fold that up. No, that's her thingamabob. I could fold up and trash one of my seniors' tests. I don't think they'll miss it. No. Okay, so I'm just using this. All right. So I showed you in class. If this piece of paper represents a big fat protein, okay, big fat polymer protein, polypeptide. I have an it. So here, let me draw my enzymes. Enzyme. Or not enzyme, amino acids. Okay. All right. My protein. amino acid, amino acid, amino acid, amino acid. All of these are amino acids, right? All bonded through peptide bonds. Beep, 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 beep. Peptide bond, peptide bond. Okay. So this big fat protein can fold up and become an enzyme. This is an enzyme, right? This is one of hundreds and thousands of different enzymes inside your body doing jobs. So it's a big fat protein in the shape of an enzyme. Every enzyme has a unique active site for its substrate, right? Perfect puzzle piece. But if your pH goes off kilter or your temperature goes off kilter, then your enzyme will unfold. And when it unfolds, it's called denaturing, right? It's not its normal structure or nature. Hey, Grant. So if it unfolds and unfolds, notice it doesn't have its active site anymore. So it's not able to do its job. It can't find its substrate and help the reaction. So when your pH is off or your temperature is off, you are in trouble. That means enzymes aren't helping your body function right now. That's why you can't stay at a high temperature. You can't have pH off in your blood or become acidotic. It's a problem. Enzymes. But notice what happens. Denaturing. Even if my enzyme completely denatures, completely loses its shape, I'm still holding my protein paper. It is still here. I have not destroyed it because, guys, denaturing does not destroy your enzymes. You can't destroy them. They just unfold. So when your temperature is back, this same piece of paper can fold back to the shape it was.
James says thank you.
Oh, okay. Yeah, sure. Good. Yeah, you're welcome. Mm-hmm. Okay, it'll fold back to its normal size or normal shape. Again, that's if homeostasis isn't working. Now let me show you what happens in competitive versus non-competitive. Two different kinds of inhibitors. One gets in the way of the active site. One gets in the way of just changing the shape so the substrate can't bind. So I'm going to turn the computer around and Gracie's going to come help me.
Oh, I am.
Yep.
I'm busy.
Too bad.
I'm trying to redo these cities because, like, I can't trust these. They look like jello.
All right. Oh, all right. So just real quick, kind of get this pointed down just a little bit. All right. So imagine the enzyme is this chair. This is my enzyme, right? It has a perfect little shape. It has a perfect little spot called the what?
Active site.
Active site, because enzymes have to be very active, right, to do their job. So active site. Gracie is my substrate. Her butt fits perfect, right? Perfect puzzle piece. Come sit in your active site, my substrate. Ding! Chemical reaction. She's done. There's the product. Okay. That is how a chemical reaction should happen. Substrate sits in the active site. Now, watch what happens when a competitive inhibitor comes around.
Gotta go sit on the enzyme.
Ding!
Ah.
And I, as a competition, sit in the active site instead and say, yo. Now, I'm not necessarily permanent. Some are permanent, and that means poison, which means death. Okay? I'm not permanent, right? This is Tylenol. This is aspirin. I will eventually get back up, go about my day, become metabolized by your body, and she can go back and do her job just like normal, right?
Sit weirdly, right, Shrima?
There you go. So competitive literally takes their seat, okay? Now get up. And I described it like, who likes to take your seat? Well, her little bro likes to take her seat all the time in the car, right?
Oh, yeah.
Now,
He's
that's
elbowing
competitive.
for it.
Non-competitive. They don't care about the active site because they have another trick up their sleeve. So come on, substrate, combine. I, as a non-competitive, I didn't do it. I, as a non-competitive, can bind back here and now change the shape of my enzyme. The shape is different, which means the active site is changed. Her butt can't fit here. Okay? So she's stuck waiting until I, as the non-competitive inhibitor, move out of the way, let it fold back, and now she can sit.
What's an example of non-competitive?
Again, your non-competitive, as far as specific examples of which ones are competitive and non-competitive, you don't need to know. But again, things like non-competitive would be drugs for HIV. People who are being treated for HIV, one of the drugs they have to take to try and keep their virus from replicating is actually a non-competitive inhibitor. So again, guys, inhibitors don't mean bad. Some of them are good. Your Tylenol, your ibuprofen, your Advil, those are good. Okay. It is your statins and stuff for cholesterol. Like pretty much all your medications are an inhibitor for some kind of enzyme. Those are good. But then you do have inhibitors that are bad. Neurotoxins from the black mamba, right?
Poison.
Cone snails, poison, cyanide, stuff like that. Those are bad and they will. Okay. Let's see.
Are there things like, oh, what are they called? Like the stuff that if you have poison and you take it to like get rid of the poison. Are those things that can like kick out the competitive?
It could. Yeah, it can. She was saying if things can kick out, like you have a poison and then you take something that gets rid of the poison, is that, yeah, like some of that stuff, it actually like causes bonding to be altered, and those chemicals that were an inhibitor can be kicked out of that active site or that enzyme. Some of the questions, she be drawing buildings. She is be drawing buildings.
They're not very good.
They're not very good. Okay. Didn't we do that, Riley? Yes, that is what y'all were doing, Toby. So Toby was a competitive inhibitor and Riley said no, but too bad Toby won out. Okay. No, she did the same thing. Yes, that's that. Yes, that is it. I didn't want to have to explain because now it's not funny. What? Okay. Why does she sit so weird? I don't know.
Is that true, Mom?
Okay, what would the dog be? Oh, did you see the dog in the camera? The dog is just another enzyme walking around. Yeah, inside the cell, pretty much. Definitely won't forget the chair.
Dog.
Good. Yeah, that's Dumb Dog. That is his name, Dumb Dog. Daddy and I discovered what we're gonna name our next dog last night. Just whenever we have another dog, we were talking about names. All right. Oh gosh, you ready? So our next dog, so we have Harley. Harley's name is Harley Boo McDoggo Butt Good. Or Dog Batto. All right. And then you've got Bo, who's just named Dumb Dog. Okay. But I'm thinking our next dog needs to be Contessa Francesca Nutella. I wanted Contessa Francesca Angelica Fruit Loop, but Daddy said no to the Fruit Loop. So when I changed it to Nutella, he was like, okay, I can do that. But Contessa Francesca Nutella, come on. Come on.
The Fruit Loop one.
I like the Fruit Loop one. I told him we're just going to get a dog so I can name it Fruit Loop. Okay. All right, guys, that really is
Your
it.
science teachers, ladies and gentlemen.
That makes sense. Looks really good. Okay. Fruit Loop is art, right? Jessica, seriously. Come on, Fruit Loop. Okay. So questions like that. I know I may be simplifying it and saying, ooh, that's it, but really it's those three things. Okay. The chart. If you can do the chart, identify the traits and structures. If you can do the graph, and if you can do your enzymes and describe things that might change them, called denaturing, such as pH, salinity, and temperature, and then how competitive and non-competitive influence. Okay. You have my approval. Oh, thanks. Nico said good. Yeah, we have his approval. So I'll just kind of, we have a few minutes because normally these go for an hour. So I'll just kind of quiz you a little bit. Obviously I can't hear you, but you'll know whether or not you're getting it right. Okay. So bam. What is that called? Gracie.
What?
What is this called?
That's a nucleic acid.
Okay, so she said nucleic acid. She's not wrong, but she's wrong. Okay. Riley said nucleic acid too. You're not wrong, but you're wrong.
Oh.
This is a piece that will help build a nucleic acid.
Monomer.
It's the monomer called a nucleotide. Timosh said, I think it's Tinmish. You're going to have to tell me how to pronounce that tomorrow, okay? But he said nucleotide. Okay. So this is the monomer that builds them up.
So monomer.
Okay. So, but you have to also be able to label it.
Oh, I can label it.
Mish. Took me a week too. Okay, Gracie is labeling the nucleotide. Riley, that doesn't help me. Okay, Gracie's going all kinds of like, hey, let's label this. All right, good job.
I don't know how to spell it.
All right, she can't spell, but that's okay. She said phosphate, sugar, base. Correct. Draw it and label it. You're good. All right.
I know phosphorus. What's the difference between phosphorus and phosphate?
Phosphorus is the individual element.
Okay.
When it binds with oxygens,
Yeah.
it becomes a phosphate.
Okay.
Which is what that is.
Okay.
Okay, T-Dog. Oh, can I just call you T-Dog? Tinmish? Tinmish? Did I say that? Tinmish? Tinam? T-name? T-name? Okay, my goodness, what an idea. Can you show the chart up so close so I can take a screenshot of it? Sure, it's all kinds of chicken scratch, but yeah. Go for it. Yeah. Uh-huh. Yep. All right. Good deal, Emily. So draw that. I don't know. What do y'all need me to quiz you on? Like, can you draw the graph?
A practice test?
Practice. I ain't giving you no practice test. Yes, you're good. Recognize, let's see. All right. So if I ask you things like, oh, here, I'll do this. I'll give you this. Oh, wrong direction. Can you quiz us on bio? Shrimon, can you quiz us on bio? Yeah. Oh, now biochem. Frickin' and frackin' child. Okay. All right, let me ask you this. If I said... Ooh, I'm going to rewrite this. Okay, I'm going to write some words down. I'm just going to write some random words down.
Dog. Cat.
And then, yes, dog, cat. Bad doggo. Can you, when I show you this, can you figure out what organic molecule they belong to?
Yes.
Just random words, okay? So let me give you this. Is that a UFO?
Yeah.
Yes.
I don't want to draw.
Okay. Do boop 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 boop. Do.
Sorry about to write my name.
I'm just throwing some stuff down, so just give me a sec. Okay. Oh
my gosh, they look so bad without the outlines. You want to see my plant and my buildings?
Oh yes, Gracie wants to share.
I can't draw
her
realistically, so I draw. Oh, it's too bright. So I draw like unrealistically.
Yeah, I don't.
There
there you go. There's her buildings.
I'm not done.
I know, it's good job, baby. All right. I don't know what some of these words. Whoever gets it first gets a bathroom pass.
I think I accidentally went at that.
Okay. So random words, but can you identify which word is associated with which organic molecule? Okay. Well, yes, Riley, I hope there's one. Okay. All right, so if you're looking at it, just take a second before I start answering. But each of these words is associated somehow to one organic molecule. Write it all down. Who's Heather?
Told you, there's a bunch of kids who are too.
No, they just all keep saying, shut up, Heather.
Oh, it's from TikTok.
Oh,
The
heavens.
musical Heather's famous sound right now.
Guys, I'm still just on the catch the whoa.
That's so old.
I know. Look who's with her. Oh, dear Lord. Wait, no, it disturbs me that y'all listen to Heather's.
Who's with her? Oh my gosh.
Stream on. Dang, diggity, dang, dang. That's, oh my gosh. Oh, I thought it was like a group of words that describes one. No, I put multiple words and you need to figure out which one's which.
Of the four?
Like a word is under the category organic compound, but which one? Okay.
Wait,
So when you see peptide, what is it? What is that associated with? And Gracie, go for it.
Protein.
Protein. Good job. Oil is associated with lipids.
Wait, we have to give them a second. Take some off.
Okay, sorry. Oh, Riley got it.
I know, you have to give them a second.
I was giving them a second.
It's delayed for them. Ooh, I forgot.
Pentagonal structure is meant for...
No, like they probably typed it like 20 seconds ago, but it doesn't show
No,
up.
Riley actually said it's not delayed at all.
Really?
Yeah. Carbs, good job.
Whoa.
Hot. Okay. Genes. If I just said genes are associated with... Okay, Toby got it. Well, Toby got part of it. Toby kind of said it. I like a second or two. Okay, so Toby said nucleotide. That's not the name of the organic compound. The organic compound is a nucleic acid. Good job, Nico. Okay. An immune system. Your immune system is associated with what organic compound? It's okay, T-Dog. Come on, give me. Come on. Immune system. What organic compound would help support our immune system as a big, like, factor? Nobody knows? Gracie?
Protein?
Protein. Good job, Riley. Yeah. He said it at the same time. Good job. Proteins. Proteins are a big factor in your immune system. Okay. All right. Glucose. Riley got it. Carbs.
Riley, quit answering.
DNA is what organic compound? Gracie?
Nucleic acid.
Nucleic acids. I refuse to be stupid.
Never
That's
apologize
Riley.
for being the smartest person in the room.
Darn right, man. Never apologize. Okay. Okay, this one?
What
About this
I'm
one?
gonna wait for Riley on.
Riley is a big boy. Riley says protein. Do we agree?
I said lit.
Jessica says protein. Ooh, Toby, you said DNA. I'm very happy you're almost trying to spell deoxyribonucleic acid.
But
using voice. Yeah, oh, Nico's like he's using voice type. Okay, guys, the nitrogen. You need to recognize this as a protein. This is an amino acid.
Okay.
Because nitrogen with these hydrogens is called an amine. And then over here is something called an acid. So this is an amino acid.
Nitrogen equals protein.
Nitrogen equals protein. All right. And then last one, enzymes are?
Lipids. Just so y'all know, I did that purposely.
What's the difference between nucleic acid and amino acid?
I wrote it down though correctly.
Good job. Enzymes are proteins, just so you have that last one. Okay, Scarlett, what's the difference between nucleic acids and amino acids? Okay. Not a whole lot, but I'm not ready for us to get there yet, but I'll go ahead and give it to you just a little bit.
Wait, what she asked?
She asked what's the difference between an amino acid and a nucleic acid? Like, why is a nucleic acid called nucleic acid? And why is an amino acid called an acid and why they sound alike?
Exactly.
Because this guy
Science
down
is
here.
weird.
Okay. Oh, so this guy down here, this nitrogenous base, this B that we call in our nucleotide, that base is called a nitrogenous base because it has nitrogen, because it's actually a derivative of an amino acid. So nucleic acids are made of a derivative of an amino acid. Ultimately, your nucleic acids have an acidic like charge or functional group to them. So, but amino acid and nucleic acid actually have a relationship. But please, please, please don't focus on that just yet. But I wanted to answer your question, Scarlett, okay? All right. I like that word. What, protein? Nucleic acid. Nitrogenous, right? Mm-hmm. Oh, trust me, dude. We've got so many different words standard to sheet 2, such as, ready for this one? Peptidoglycan.
I've heard that word before.
Peptidoglycan, yes. And if you think about it, you actually already know what it is in a way based on the words in it. Peptidoglycan. Okay. Lumen Omni Convert. Darn right. Okay. So those were words. So close, Riley. So close to spelling it. Very, very close. Okay. Supercalifragilistic. Yes. So yes, if you can associate those words, then that means you're at least making the connection on like bonds and shape and examples to the organic compounds. Good job, guys. And then draw the enzymes, right?
I give up on this piece of art. I don't like it.
Oh my God.
Buildings, go away. Delete.
There you go, Riley. Peptidoglycan. Peptide glycan. So it's a protein sugar. Ooh. All right. What questions can I answer? I mean, do the chart. I gave you some practice words to look at, right? Draw the enzyme and label it. It's active site, substrates, products. Know where competitive, non-competitive go, right? Competitive competes for the seat. Non-competitive changes the seat. And then be able to do your graphs and draw the enzymes on there.
There's no such thing as carbon dioxygen.
No. So an active site is where the substrates bond with enzyme. Yes. Think about enzymes are very active, so they have to have an active site to pull the substrates in. Ridiculous. I kissed a girl and I liked it. Good job, Shremon. I'm glad. Katy Perry would be very proud of you. Mm-hmm. Okay, can you give us answers? No. I'm feeling no. Yes, enzymes. Yay, yeah. That's an answer. Okay. I don't know. Any questions? Any actual questions? Y'all just need to be able to do those things. I'll be there in the morning if you have any last minute things. Y'all can. Lunch isn't really an option because I have duty on Thursdays. So you have to come in the morning if you have any last minute stuff. If you do get there in time, then I definitely recommend, grabbing a marker, getting on the board, and drawing the stuff. And if you come in with people, then it's helpful because then the other people can kind of help you or you can help them. And trust me, that is what studying needs to look like. That's how you get better at it. She said duty. Oh, my dear Lord. Catalase's job was to react with hydrogen peroxide. Like that was the lab we did yesterday, was the H2O2. Don't be a meanie. Okay. Anything else? We're right there at 1 hour. Okay, good for you, Gracie. Anything? I'm confused over nucleic acids and DNA. Nucleic acids are your organic compound, right? It's in the chart. And an example of a nucleic acid is DNA. Another example is RNA and ATP.
Riley, the girl Riley wants, because she can't comment, she wants to ask you if you can dab on camera.
Heavens to Betsy. I reported his kissing comment. Darn right. He's like inappropriate. I mean, sheesh. Okay, I was going to say, you just jelly. Why are functional groups? All right, so Scarlett, reply back. Let me know if you still have a question over it. What I answered about nucleic acids and DNA. Why are the functional groups of carbohydrate? What are the functional groups of carbohydrates, lipids, proteins, nucleic acids? Okay, so one thing real quick. So Ryan, you are in Martin's class. I'll answer your question, no problem. But guys, Martin's class goes over functional groups. I didn't go over functional groups. It's one of those, like, she went over that, but I went over phospholipids and peptide bonds. You know, it's like there's little extras that teachers like to fit in as their own personal thing. So Ryan, I will go over that with you in just a second. What is made by joining together amino acid subunits? Amino acids are the monomer of what, Grace?
Amino acids are the monomer of proteins.
Proteins. So when you bind amino acids together by the peptide bond, you build a protein. Okay, Scarlett, be sure you answer and let me know that you're good on your question. About the nucleic acids are the polymer and DNA is an example. Like, that's the organic compound type. Okay, functional groups, Ryan, of your carbs and stuff. So when we're looking at our carbs, let me write this. Okay, carbs are going to typically only end up having things like, oops, I did not draw that well. Carbs are typically going to only have OH, hydroxyls, hanging off of their pentagonal or hexagonal structure. They'll have hydroxyls. Your lipids are going to end up having, and I don't know how many functional groups she went over. I don't do functional groups until I get into, unless you're in my AP class, because there's a lot. Your lipids are going to have two things about them. And if these aren't mentioned, then ignore them because this is beyond. Lipids are going to have carboxyl and methyls. The carboxyl helps it to bind to glycerol, but then the methyl functional group is actually what makes lipids nonpolar. Okay. Proteins are going to have your amine. Proteins have the amine. That's the nitrogen group that you need to recognize. That's the amino. But then they also have the carboxyl as well. So when you see a nitrogen structure for for amino acid, when you see this, there's the functional group amine and the functional group carboxyl. And that's what a protein has. Now, proteins can actually have a lot of functional groups because down here, this R placeholder, that R can become anything. That's what makes every amino acid in our world unique. There are over 500 amino acids in our world, and they are all unique because of that R, whatever that R is. So that can be a different functional group.
Like the X and Y.
And then your nucleic acids, your nucleic acids have a little bit of everything. First, they have a phosphate. They have a phosphate group. Because of the sugar, they're going to have a hydroxyl. Because sugars are carbs, right? They have a hydroxyl. And because of the nitrogen base, they're going to have an amine and a carboxyl again. Okay. So the nucleic acid will have a little bit of everything. Again, Ryan, I'm not sure which functional group she goes over or not. Hydroxyl is the most common one that you see. But the amine and the carboxyl are the important ones to know for proteins. Okay, let me catch up on a couple of questions. Hold on just a second. There are quite a few questions I need to go back. Okay, what are the functional groups? Ryan, I hope I answered your question. Let me know. Morgan, amino acids join together to make proteins by the peptide bond. So enzymes are the catalysts which speed up the process of time it takes for the reaction. Yes, Nico. Enzymes are an example of a catalyst. A catalyst is anything that can speed up a reaction. You can be stirring your sugar, and that is actually a catalyst. You can put your food in the oven to get it done faster. That heat is a catalyst. What makes up nucleotides? Riley was answering that. PBS, phosphate-based sugar. You have to be able to draw it. Just think about it. Man, where'd you learn that? I learned that on the Learning Channel, PBS. Phosphate-based sugar. Phosphates are always circles. The pentagon is always a sugar, and this is a box base. Scarlet, well, technically yes, but in a different order. Oh, sorry, I skipped that part. Nucleotides are PBS, right? No, Riley. Yes. I see what you're saying. PBS is just the way to remember it. But yes, guys, I need you to put it is in this order. A phosphate, a sugar, and then the base. Like this is the actual order. PBS, just remember it. That's how you remember it. Good. Thank you, Riley. Hold up. Oh, there's so many. Hold on. Hold up. What is the difference between amino acids and nucleotides? Again, the amino acid looks like this. Let me find what just drew that. Okay. Scarlett, this is an amino acid. This is what builds a protein. This is a nucleotide that builds a nucleic acid. They're very different structures. They just have this guy is part of what made up that box there, the base. It's part of that. So really, this nucleotide's more evolved. Like it has a lot more to it. So R is basically an X in math. Yes, Jack. Replace the R with whatever needs to go there. It can be any element. It might be a carbon. It might be a phosphate. It might be a sulfur. It's anything. That's what makes all 500 unique. How many amino acids are in the human body? 20. We have a total of 20. You make 11. You have to eat 9. We have to get 9 from our food that we take in. Can you put up the close up? Can you put that up close so I can get a screenshot? Which part? What do you need? T-Dog, I don't know which thing you're needing. Are you needing the functional group thing? I don't know. You need to tell me what you're needing, T-Dog. I don't know. Amino acids are the monomers of proteins. Nucleotides are not. Good job, Riley. Thank you for answering her. Jessica, amino acids make up. Thank you for answering her. Sorry, I'm trying to catch up on all these. Ryan, good. I hope I answered your question. Okay, cool. Scarlett says 11 are generated RA. You need to ingest. Riley, you're doing great. What goes into the chemical reaction what goes into a chemical reaction and comes out? Reactants. Your question doesn't make sense. A reactant doesn't come out. Reactants are what gets started in a chemical reaction. Okay. The chemical reactions, A plus B will make C. A and B are your reactants. C is your product. But then some reactions, C can then be broken to make A and B again. But still, C is your reactant and A and B are your products in this case. It's the difference of building anabolic versus breaking catabolic. So, Toby, I'm not sure how you phrase that question. Could you give us a checklist of the stuff we should know before the test? I just did.
Yeah, but can I write it down?
No. You need to do the chart. You need to be able to label and draw the graph. You need to be able to label and draw the enzymes and substrates and active site. That is your stuff. Or are reactants first and come out something? Reactants are what have to react together to form a product. Just like Dax said it in tutoring when he was talking to somebody. He was like, think about it as math. The product is always what's at the end, right? That is your product. Because reactants have to react first. The product comes after. Okay, 1 chart, 2 graphs. Thank you, Riley. Somebody's paying attention. I understand now. Good. All right, thanks, Riley. Good. Yes, yes. Can you help with the chart on the test review? The chart on the test review is the chart that we've been doing. It's this. It's that chart. You just need to fill in those last these 2 pieces. Okay? Just fill that in. I got the screenshot. Thanks to function group. Just had to rewind. Okay. All right. Finally caught up on all the comments.
Did you?
I did. Are we done?
Probably all
we
going back
done?
and rewatching.
I know y'all are all going to go back and rewatch it. It's fine, whatever. I try and keep it at an hour because whoo, that's a lot. Whoo. All right, hit me up in the morning. Okay, y'all still have questions or whatnot. Anything else? Give you another about 10 seconds to let me know if you have another question. 10. Toby says thank you. Riley says thanks. I think that's it. All right, thanks for doing this. Yep. And Riley McAuliffe said, please dab. Ding. Is that good enough? Don't forget the task sheet and tic-tac-toe. Nico, you're amazing. Task sheet and tic-tac-toes. Y'all technically should have already finished the task sheet because we did that one day in class. Okay. No, don't. By the way, thank you so much. Yes, you're good. All right. Bye, guys. Yep. Talk to you later. Oh, ask me one question in teaching is really... Yes.
What?
Riley said this was really just me asking one question and teaching everyone else. All right. Bye, guys.